I have some comments about how, you know, things that I hope that students can take away in terms of thinking about your own lives and your own journeys in science. So I'm a developmental biologist, and I just spilled water all over myself, so yay for poise, right? So this is what I'm showing you here is a cross-section of a tadpole brain. And I'm mostly showing you this picture because it's kind of pretty. But let me tell you a little bit about the kind of science that I do and how I got started with this. So I study embryonic development, and this is a picture of a human embryo at about 7 weeks. And what you see is something that looks like it will grow up to be a baby. It's got arms and legs. You can see where the fingers are going to form. It's got a beating heart. It's got an eye. It's got a magnificent start to making its brain. And it's 1/4 inch long. And here's what it looked like just a few weeks earlier. Looked like a ball of cells. And I was captivated by the question of how you go from being this ball of cells to being an embryo when I was an undergraduate. And I kind of got that question in my teeth, and I've never let go of it. And we focus it down more, and we think about developmental biology. And I'm sure some of you have seen this before. I'm going to ask you to just humor me for a minute while I tell you stuff you already know. So we start out with a fertilized egg. It's a zygote, and that one cell can give rise to all the cell types of the body. It doesn't do that directly. It goes through a phase where we have a lot of cleavage divisions, and what we're looking at up here is a frog embryo, but these basic principles apply to lots and lots of embryos, to basically all embryos. Both plant and animal, incidentally, but I haven't thought about plant development in a long time. So this fertilized egg will divide and divide and divide, and it will eventually form a kind of a mass of cells. And in the frog, that mass of cells is a ball of cells. In bird embryos or mammalian embryos, it's a sort of a flatter surface of cells. But you have this mass of cells, and then it undergoes a kind of a large-scale rearrangement that we call gastrulation, and you set up 3 layers. You all heard this back in high school, right? You set up 3 layers. The endoderm is this most internal layer, and that's going to give rise to the respiratory system and the gastrointestinal system. There's an outer layer of the ectoderm, and that's going to give rise to the skin and the central nervous system and a bunch of related tissues in the body that are derived from the neural crest. And then there's this intermediate layer that gives rise to things like our skeleton, our muscular tissue, the red blood cells, the kidneys, the somatic tissues of the gonad, and some other cell types. So there's a kind of a step-by-step process where you segregate into these layers. These layers can then give rise to a set of cell types, like ectoderm cells can only give rise to these cells. They're not going to give rise to endoderm types of cells. There's this kind of restriction in the developmental capacity that cells have to give rise to new cell types. And ultimately, they commit to forming a specific cell type, such as the neurons, and they lose the capacity to form other cell types. That's basically how developmental biology works. And this involves changes in gene activity, and it involves, and those changes in gene activity are governed by components that are intrinsic to the fertilized egg. Some of them are down at the bottom and some of them are up at the top. But also there's a process of communication between cells that starts very early and continues on. And even within and between all of these layers, cells are constantly talking to one another and trying to understand how they talk to one another, how they interpret what they are hearing from their neighboring cells, how that brings about changes in gene expression. Those have been the questions that I have stayed with throughout my graduate training, when I worked on heart development, and then when I was a postdoc at UC Berkeley, I moved to thinking about development of the central nervous system, and then in my own independent laboratory where we've worked primarily on early neural development. And I study these questions in frogs, as it turns out. I have, when I was an undergraduate, I worked on Drosophila, fruit fly. I've had the opportunity to work on a number of other organisms, including bacteria and mammalian cultured cells and slime molds and chick embryos and sea urchin embryos.
Dr.
And yes.
Is there a specific reason why you chose this species of frog in particular? Because I know we choose the Drosophila because they reproduce very quickly, and that's why they're advantageous to study genetics. Why this species in particular for your research?
That's a great question, and I am just about to tell you, to explain that. That's a really important point. This species is a commonly used, we call them model systems. Another word for a model system is an animal that you can grow in the lab. It's easy to raise in the lab. And I'll talk a little bit more in a minute. I have a slide that addresses your very question. But this is a picture of this. This is what frog sex looks like. Fertilization is external, and you wind up with this mass of oocytes that are happily color-coded. So what we call the animal pole is dark, and the vegetal pole is lighter. And as the embryo begins to cleave, you see, we're looking at the animal pole here, and you can see where the cells are forming. This is a somewhat older embryo. Now I'm going to flip the embryo over, and we're looking at the vegetal region. And this is where that process of tissue rearrangement called gastrulation begins. And an awful lot is happening at gastrulation. There's a kind of a conversational storm happening here. There's lots of communication. And at the end of gastrulation, the embryo has kind of organized itself spatially, and we begin the process of forming the central nervous system. This keyhole-shaped thing is going to roll up and form the neural tube, and the neural tube goes on to form the brain and spinal cord, and this is what we call a tailbud embryo. And at this point, the organ systems are all kind of in place, and they're starting to form. And then we have larval development. So you're all familiar with tadpoles because you see them after, a couple weeks after a rainstorm. You can see tadpoles in ponds and puddles. Interestingly, as the tadpole begins metamorphosis, there's a whole burst of a kind of a second round of neural development, which I may talk about a little bit later. So there's a kind of another period where there's a lot of action going on in terms of forming new capabilities in the nervous system. So to get back to your question, why frogs? I mean, why this species of frog, but also why frogs in general? Why would you work on frogs when, as I think about it, you could do slower and more expensive studies using the mouse embryo? There are a bunch of folks that work with mice. There are a bunch of folks that study development in Drosophila or zebrafish. And also there's a nematode worm, Caenorhabditis elegans, that people use to study development. Some people use chick embryos to study development. And we all talk to each other, but we all secretly prefer our own system because we like to we're looking for different things. So for me, the most important things about this frog, in addition to the fact that it's a well-established laboratory animal, it's originally from South Africa, but it was brought to the United Kingdom in the 30s, and people started to do research with it. What we now know is that the frog genome is very, very similar to the human genome. The genes that regulate early development in frogs and mammals are basically identical. Genes that regulate cell division and the emergence of form, morphogenesis, fundamentally identical. There are direct correspondences for genes in mice versus genes in frogs for something like 85% of the genes in our genome, I think. So lots of kind of fundamental similarities, even though obviously the morphologies and the life cycles are very different. We can obtain hundreds of synchronously developing embryos very easily. The frogs themselves live 10 to 15 years, 20 years sometimes. And so I can keep a colony of frogs in the basement of my building and just grow up the embryos, and when we need them. So we generate embryos probably 4 or 5 days a week, and we can culture them. We can do both functional studies to understand how a gene specifically functions. We can also do mechanistic studies, which often involve biochemistry. And those of you who've had to do some biochemistry know that if you're going to try to do some biochemistry, you have to have a lot of starting material. And that's where working with frogs is an advantage, because we can get so much material. If people who work on mouse embryos generally don't do mechanistic studies in vivo, because it's every mouse embryo is precious. They're developing internally, and if you're collecting mouse embryos, you have to dissect out the uterus and then remove the embryos, and there's usually maybe 8 to 10 in a given female. So it's really appropriate for very different kinds of experiments. And so I have a bunch of friends in other institutions who they have mouse projects and frog projects, and they'll do the early stages of a study in the frog and then port their results into the mouse system. But I work only on frogs. I'm really happy. The kinds of experiments I like to do are easy to do in frog, and we have opportunities for rapid and cost-effective studies of gene function. I do think, though, that to really understand developmental biology, it's important to draw on work from a broad range of systems. And I try to pay attention to what's happening, not just in studies using my system, but also mouse and various other organisms as well, certainly Drosophila. Does that answer your question?
Yes, thank you, Dr. Sater.
Sure. So I want to shift gears a little bit and tell you a little bit about myself. I teach at the University of Houston, and when I compare myself to my students on our campus, I feel like I had some advantages that most of my students don't have. And one of those advantages is I'm 500 years old, which means that I went to college at a time when college wasn't nearly as expensive as it is now. I didn't have to take out student loans. I grew up in Northern California, about an hour north of San Francisco. And in California, the state had put a ton of money into developing the University of California system. And so I was able to go to college, and it just wasn't that expensive. And my parents had worked their way through college, and so they didn't want their kids to work through college. So I could just really focus on my schoolwork. And that's something that most of the students in my classes at UH do not have that privilege. So I feel very humbled by that, because I know that I did college at a lower degree of difficulty than most of my students, and probably many of you as well. I only had to worry about kind of, getting my schoolwork done and keeping my own self together, and I wasn't worried about taking care of my family, and I wasn't worried about getting to work or anything like that. So those are sort of tangible, obvious benefits. But I had some intangible benefits as well. I went to a high school that was terrible. I shouldn't say it was terrible. It was academically not that great. It was in a somewhat, so there were not things like advanced placement classes at my school. And so at a certain point, when I finished my junior year, I realized that I had an opportunity to just skip my senior year and go to community college. There was a special test you could take that would allow you to do that. So I didn't graduate from high school, which means I don't have a high school class rank, and nobody, which becomes this thing that you have to kind of deal with. I went to the University of California at Santa Cruz, and back then, UC Santa Cruz didn't have grades. The Fighting Banana Slugs, go Banana Slugs. We didn't have grades, so I didn't have an undergraduate GPA. We had what was called a narrative evaluation system, in which instead of a grade, your instructor wrote a paragraph about your performance, which I'm sure seemed like a great idea at the time, but after I graduated, a few years after I graduated, the faculty at UC Santa Cruz were like, yeah, we need to not do this anymore. This is really time consuming. So I didn't have that pressure of maintaining a GPA. That's not how we were evaluated. I didn't have a lot of parental expectations about going to medical school or going to law school or getting a job right out of college or setting myself up in a career. And here we have a picture of my dad. My dad was an all-time fantastic dad. And he was a civil rights attorney, and then, excuse me, he was a judge, a county judge, and for a while he taught constitutional law, kind of on the side because they needed the local law school needed someone to do that. But before he went to law, and he went to law school at 32, and you might wonder, well, what was he doing for the 10 years before he went to law school? And for most of those 10 years before he went to law school, he was an actor in Manhattan. So my parents don't have any credibility at all for any kind of question about, when are you going to decide what to do with your life? I would just look at them and say, you have no credibility. You guys, my parents didn't figure out what they wanted to do until they were in their late 20s. And so these things, I see my students at UH, and I see how competitive and challenged and kind of this whole thing of, a lot of the students in my classes want to go to medical school, and they're tracking their GPAs really carefully, and they're trying to build a portfolio that will get them into medical school. And that seems so exhausting and distracting. And I know that for students that are really enmeshed in that, you've got to figure out how to do it because that's how the system works. But I think sometimes it can distract you from figuring out what you are most interested in, what your strengths are, how you want to figure out what you want to do, because those are hoops that you wind up having to jump through. Now, I had to jump through different hoops, but I think it's important to have some breathing room to figure out what your passion is, what you really want to do. And with my students at UH, that's what I try to talk to them about. So when I was an undergraduate, I had the opportunity to spend a whole term at a marine laboratory, Bodega Marine Laboratory. It's on the California coast. And I generally think about my initiation into science and a lot of my early time as, you know, as an undergraduate and then in graduate school, as my life in 4 marine labs. And so that's what I'm going to show you. And I was at Bodega Marine Lab for a whole term. I loved it. I just loved every minute of it. We'd be out in the intertidal taking notes and looking at animals and turning over rocks and getting really wet and asking lots of science questions and thinking about how do you figure out the answer to these questions. And I just thought, wow, this is something I really love doing. And I got to know some of the graduate students there. I wound up staying on for the summer, so I was there for a total of like 7 or 8 months, and got to know the grad students. From them, I made the erroneous conclusion that being in grad school meant that you spend a lot of time going out for low tide when you can look at the animals you're trying to observe. And you cook Chinese food at home and you drink beer. And I thought, I'm on board with all of those things. I can learn to cook Chinese food. That sounds like fun. That turns out not to be a good, accurate representation of graduate school. But I went back to UC Santa Cruz and I started working in a research lab, working on Drosophila. And that was great. That was a really good experience. And then the day after I graduated from college, I went off to Friday Harbor Laboratories, which is northwest of Seattle. And this is a picture of Friday Harbor Laboratories. It's on San Juan Island. And spent the summer at a marine lab, took some classes, learned about this animal here is called a Staromedusan, and it has a very interesting complex life cycle. So it's related to jellyfish, and it has a phase of its life cycle where it's a jellyfish, and it has a phase of its life cycle where it looks like this. And again, this is back to why developmental biology is so amazing. This doesn't really look anything like a jellyfish, and it's sitting down on the bottom. It's sitting down on a rock as opposed to swimming in the water. So this very much stoked my desire to continue in developmental biology. And the year after that, I worked as a research technician, and I applied to grad school and wound up at Stanford. And spent a year on the main campus at Stanford, and then spent the second year at Hopkins Marine Station, which is, again, West Coast, California. This is at the southern end of Monterey Bay. And so if any of you have read John Steinbeck's Cannery Row, Hopkins Marine Station is right down the street from Cannery Row. And this was a really interesting place to work. It was also a lonely place to work because there were very few graduate students there. There were a bunch of people who were like staff scientists, and they were very settled, and they were older than me. And so I spent a year kind of reading and thinking and doing experiments, and eventually realized that this was not the science I wanted to do. Because somewhere along the way, I realized that vertebrate embryos are offered a lot more opportunities to study the kind of cell communication that I'd gotten really interested in. And I wanted to work on vertebrate embryos, and there wasn't anyone at Hopkins Marine Station who worked on vertebrate embryos, and there wasn't really anyone at Stanford who had an active research program on vertebrate development. And I started thinking about, did I want to just stay at Stanford and just graduate and then pursue my research interest in a postdoc? And I decided, I talked with a friend of mine who was a postdoc at Berkeley, and he said, you know, life is short. You should go find the training that you want to get. And so I left Stanford. I took a master's and I moved to the University of Texas at Austin. And I can't tell you how many people told me I was crazy. Hey, you could get a PhD from Stanford. That's got to be worth it. And I thought, I really want to do the science I really want to do. And so that's the choice I made. And I have not regretted it. It was the right thing for me to do. So I went to UT Austin. I worked on heart development, started out working in chick embryos, moved pretty quickly to the frog embryo. I was there for 4 years, and during those 4 years, I spent 2 summers at Woods Hole at the Marine Biological Laboratory. Now we get off the West Coast and we go to Massachusetts. Woods Hole, Massachusetts is right at the edge of the southern edge of Cape Cod. If you've been up to Boston, it's about a 2-hour drive down from Boston. And it is one of the oldest marine laboratories in the United States, or actually it's one of the oldest in the world. It was founded in 1888, and it's just this wonderful little tiny, it's a big marine lab surrounded by a very tiny community right at the edge of this saltwater pond, which is called Eel Pond. And I spent 2 summers there taking the embryology course and then being a teaching assistant in the embryology course. And I've gone back every few years ever since for workshops or meetings, or there's a big research facility there for people who work on Xenopus, and it's available for those of us who want to come and just work for a few weeks. So this kind of was a great way to go through my 20s, spending summers at marine laboratories. At a certain point, you realize you've got to buckle down and actually finish your PhD and do the writing. And I got my PhD, and then I went back to UC Berkeley, and I postdoced with 2 people, and I worked on a project very much of my own developing, and eventually I landed at the University of Houston to set up my own research lab. And some years after that, I was at a conference at the University of Virginia, and with a bunch of people who worked on Xenopus, and we were all becoming increasingly aware. So the Human Genome Project was underway, and it was the first draft of the human genome was about to be released. And we were aware, as people who work on Xenopus, that it was going to be important to have some genetics and some genomic resources for our species. And so at this conference, we talked about how to do that. There's maybe, excuse me, maybe 100 people in the room. And people were talking about how they were going to do stuff. And I looked around the room and I thought, there's not a single person in this room that actually knows how to do any of the things we're talking about here. And so my bright idea was to come back to Houston and see if I could talk a human geneticist I knew into collaborating on this project. Because how hard can it be? The answer to that question is very, very, very hard. It will take 10 years to do what we did. But so this is a picture of the Dean of Natural Sciences and Mathematics at the University of Houston. He's my boss. And he's also my friend. And I came back from that meeting and I said, Dan, let's go out to lunch. I want to pitch an idea to you. And so I took him out to lunch. We ordered wine. I mean, who drinks wine at lunch, right? And I basically said, we need your expertise as a human geneticist to develop genetics in Xenopus. And I talked him into it. And then I went back to my science community and I said, hey, I just got a human geneticist on board, so we'll make the genetic map. And everybody was like, oh, thank God, go do that. And then we got funding from the National Institutes of Health to do this genetic map. And I was absolutely paralyzed with fear for about 3 days because I didn't know how to do this. And I had just told people I was going to get it done. And we'd written a grant, but I felt so viscerally outside of my comfort zone. And I was really scared. But we broke it down into chunks. And what I've learned over the years is that Dan Wells' strengths and mine are very complementary. And so the things that I do well are things he's happy to just let me do well. And the things he does well are things that I don't do well. So we work well together. We spent 10 years working on this project. And we worked a lot with a bunch of people. And a bunch of them are shown in this slide. And many of these people are my friends, but do you see a pattern here? We have really a lot of guys, lovely guys, really wonderful people, but they are not only guys. Most of them are what I affectionately call pasty-faced white guys. Mustafa here, his family's from Pakistan. Dan's family is from Iran, but the rest of these folks are pretty Caucasian. And this is, so for 8 years, we would meet twice a year, us and sort of a larger group. This was the core group. And the meetings would alternate between being a giant phone call, conference call, or in person. And every time we met in person, there would be two women in the room, and I would be one of them. And the other would be, it was never the same woman twice. It would be somebody who had been sent from one of the genome centers that was working on the project. And she would not, my boss can't make it, so I'm here, kind of thing. And so I was the only woman on this project for a long, long time. And to be honest, it was fine. I mean, in terms of what is it like to be the only woman working on a project with a bunch of guys, I did not personally find that experience problematic. These guys were easy to work with. We got along fine. They were very supportive. Is it a good thing that for the community that this be the group that is doing this? No. We need to be, we can't afford to be rooms full of pasty-faced white guys. And I'll come back to this point. We need to be bringing everyone in. We need all the voices. So, but this is my community. These are the people who stepped up to work on developing genetic and genomic resources. Now, many years later, we have these resources. This is one of the things that we did. We made a genetic map, and then we lined it up on the chromosomes. And I'm showing you this because it's, again, it's kind of visually more interesting than most genetic maps are. And it was a huge, enormous amount of work that I would never have envisioned myself leading or co-leading. And it required a lot of conversations with people where I didn't feel like I was communicating very effectively. It really stretched me in many, many ways. But I decided that I had stood up in front of a room full of people and said, I will get this done. And so I had to get it done. I had to keep my word. The Genome Assembly itself was published in 2010. This is like a giant paper. It made the cover of Science, which was very satisfying. That is not a tadpole. doing yoga. That is a tadpole swimming upside down, basically. And so the Genome Assembly was published. The following year, our genetic map was published. These were giant projects that involved lots and lots of people. And one of the things that I really took away from this experience is that collaboration is good. Collaboration is essential. And we should all be learning to collaborate, and we should all be teaching the people we teach to collaborate as well. So that's the rationale behind all those super annoying teamwork assignments that you get. There is a point to them. The most interesting stuff that I am going to do in science will be done with other people. The most interesting stuff that Jay-Z does in music will be done with other people. It is quite likely that the most interesting work that any of you do in your lives will be done collaboratively. And so we all need to develop our collaborative skills. One of the outcomes of that project was that I wound up getting asked to come teach a course at Cold Spring Harbor Laboratory, which is not a marine laboratory. It's actually a molecular biology laboratory. And it's on Long Island. It's about 45 minutes out of New York. And I was asked to teach the Cell Biology of Development course. And I taught that course for 4 years. I'd go up and spend 2 weeks at Cold Spring Harbor, teaching up here, this building here. So I'd sleep in this building here, and I'd teach in this building over here, which was just really convenient. What they didn't tell me when they asked me to do the course is that, so the class is up here. This was a little, this is the lab over here. This window is a little kind of library area. And just outside that library area is Jim Watson's office. This is Jim Watson of Watson and Crick. And he is like 500,000 years old at this point. And he had to walk through our space to get to his office. And so we would run the class. We'd be in the lab from like 8:30 in the morning until about 10:30 at night. And then we'd get out guitars and sit in the library and play music and sing and drink beer till 2 in the morning. And then we'd run around and clean up all the beer cans so that when Jim Watson walked in the next day, he wasn't tripping over, you know, our amplifiers and beer cans. This is also my friend Takuya Nakayama, who came and helped me with the course, who's just a wonderful person. Jim Watson, a very much less than wonderful person, a terrific scientist, but he really continues to believe that science is for pasty-faced white guys. And we would start the class with a little reception for the students. And one year we were having this reception, and I went into the lab to set something up for the next day. And a half hour later, some of the students came in and they were very upset and they wanted to talk to me. And Jim Watson had said to them, "Women get distracted by their desire to have children." And they were just incensed that he would say such a thing, because it's kind of a ridiculous thing to say, right? And so I listened to them, and then I said, "I'm sorry, what did you say?" "I was momentarily distracted by my desire to have children." "Oh, wait, I already did that." So we tried to turn it into a joke. But he's, you know, we have this, he's somebody you've all heard about since like middle school. And we have this narrative about our heroes in science, and he's still a hero in science. He's still, his accomplishments changed the way we think about biology. And at the same time, he is somebody whose personal beliefs are antithetical to what science needs to do in order to grow, which is to bring in everyone. So this is the sort of difficulty that we have with a lot of people at this point. So this is the instructor team that we put together to come in. Like, I was there for the whole time, and my friend Jerry up here was there for the whole time. We were co-directing this, and then we would have all these other folks come in, and you're seeing the pattern continue, aren't you? Again, these people are my friends, but, you know, Hytham's family's from Egypt, but pretty much everybody else is a pasty-faced European originating white guy. And I am really embarrassed to say that I didn't see that. The last year I taught the class, I brought in the person who was the team that was going to replace us, as like Jerry and I had finished our time teaching this course, and we were bringing in our replacements. And our replacements included Mustafa Kokra, but also Karen Liu. And Karen looked at the list of people that I brought in, and she said, "These are all men." And I went, "Oh, wow, you're right. I hadn't even seen it," which is really embarrassing and appalling. But she helped me to shift things. So the following year, like, there was a year that we taught the course together, we brought in a broader range of people. So sometimes, you know, the moral of that story is blind spots, and we all have them. When someone's pointing out yours, it's good to listen. So finished up Cold Spring Harbor, and I was like, okay, I'm not participating in these kind of large projects outside. I just want to focus on my own lab. And I started 2 new research projects. This was in 2015. We started working on the origins of glial cells, specifically astrocytes. And there's a lot of reasons for astrocytes do a number of things developmentally that make them quite unusual. And this also becomes relevant because these astroglial cells are often the source of brain tumors such as gliomas or glioblastomas, and these are the most lethal brain tumors, and we don't have good treatments for them. So again, can we develop Xenopus as a model for thinking about glial development and trying to understand how they can shift from a stable differentiated state to a state where they're proliferating? So I was starting to do that, and then one day I got called into the dean's office, and he said, what would it take to get you to be chair of the department? And I kind of looked at him and said, what are you talking about? And we were undergoing a leadership transition, and he had, unbeknownst to me, been asking people in my department if I would be someone that people would find acceptable as a leader. And I think everybody was surprised to be asked, because I was on record as saying I never wanted to do that. But people were like, yeah, I guess she'd be okay. And so that led to me becoming department chair, which I did in 2015. And we're a really big department. There are 50 faculty in this department. We have 2,500 majors. It's a lot of responsibility. It's kind of like having a second full-time job. But I have learned so much doing this. And I want to talk to you in this last bit about leadership, which is something I didn't understand at all when I became department chair. And I went and I had leadership training, and I did some reading. And I've come to see leadership as something that is a practice. And this is a quote from someone named Ron Heifetz. Leadership is a practice. It's not a position. And we can all practice leadership irrespective of what we are doing and what role we play. Leadership is about caring about a situation and about the people in that situation, and then assisting people in directing their efforts to meet the challenge, solve the problem, adapt to change, whatever is in front of you. That you can do as a faculty member, you can do that as a student. You don't have to have the title. And I want to encourage each of you to, excuse me, to think about where in your life there are opportunities for leadership. What are the things you feel passionate about, and how can you assist the people around you in resolving those issues or advancing that knowledge, or whatever it is that speaks to you. I think about how people develop their leadership skills every day. I work with the faculty in my department to help them develop leadership skills. I'm trying to mentor a lot of people in developing their leadership capabilities. And I myself am mentored by a number of people. I am very grateful to my mentors for their help in, their assistance in helping me grow. There are just a whole lot of dimensions to being a leader. There's listening. We think about leadership and you think about, well, you're the person who's kind of imposing your direction on a situation. And I think those of you who've been in leadership situations know that that approach doesn't work very well. You need to listen to where people are. You need to show compassion and good judgment. And probably the most important quality you need to show is integrity. And take a minute and think about what that word means to you. And I'd be really interested if somebody wants to speak up and say what they associate integrity, what integrity means to them, or put it in the chat. Let's just take a minute to think about integrity. Thoughts on integrity?
It's doing the right thing when no one's looking.
Yes, it is very much that. You are anticipating my next slide. That's wonderful. It's doing the right thing for your, because you need to do the right thing because it's who you are. Yes, Raina Taylor, absolutely. It's keeping your agreements with people. It's holding yourself accountable. Being able to be a good person because it's who you are. It's your value as a person. I love these. These are standing by your morals. These are wonderful things. And I think we have, in our current society, setting 2020 aside, right? This year has been just enormously challenging for people. Even before then, things have been so competitive and so difficult and so challenging that I think that we as faculty have foregrounded, here's what you need to do to pass the class. Here's what you need to do to get an A. Here's what you need to learn. Here's what you need to review. And we've placed a lot of emphasis on lining on a bunch of academic achievement hoops. And it's not that the hoops are going to go away, I'm sorry to say, but I think that integrity is so fundamental to success. I think about the opportunities that I've had have come, yes, like all of you, we are bright, we are hardworking. I think that the opportunities that I've had have come as much because of my integrity as they have because I'm bright and hardworking, right? People, it's can you establish trust with people? And I want to encourage all of you to reflect on that sometime in the next day or two, and then kind of carry that with you. Like, that's the core of who I am. I'm not my MCAT score. I'm not my GPA. I'm not my class ranking. I am more than that. And part of what I am is my integrity. So just to anticipate your answer, here's a quote from a guy from the CIA, which is an interesting place to be talking about integrity. In the leadership course, what is integrity? Doing what is right even when no one is looking. And Alex Finley says, no, integrity is doing what's right even when everyone is looking and they're telling you not to do it. Hopefully none of you are going to find yourselves in that position anytime in the near future, but as you get older, you may encounter situations like that. And I think that this is something that all of us can kind of engage with. So I'm going to wrap up finally. I've droned on and on. There are 3 things I want you to remember. Don't let other people define you. You have strengths, you have interests, you get to define those things. Second thing I want you to remember, leadership is a practice, and you can start practicing it anytime you want. And many of you, I know, already do. It's already part of who you are. And the third thing is, figure out what integrity means to you. Excuse me. And then strive for that. And I will leave you with 2 slides. Does anybody recognize this? This is a slide you would have seen about 4 years ago. This is the negotiating team for the Iran deal. And it's a whole bunch of pasty-faced white guys and Federica Mogherini representing the European Commission. And she's the only gal in the room. And I want to say that I really want rooms to not look like that anymore. This is what a room should look like. This is the room. It's a little out of focus, but this is the kind of room that I want all of us to be in the future. This is last year's SOCNAS conference that was held in Hawaii, and which is not a great place. I mean, the people who went had a great time. Holding it in Hawaii became really challenging for a lot of people, but we want everyone up on that stage. I want each of you, excuse me, up on that stage. And I will stop there. And I will stop sharing my screen.
That was a great presentation, Dr. Sater. Thank you so much. Thank you, everybody.
Oh, my pleasure.
Yeah.
Thank you, Dr. Sater.
Yes. Any questions, anyone? Chat, or you can speak up.
I have one.
Sure.
So you encountered Dr. Watson, James Watson, the half Watson and Crick.
Yep.
How does he react when people bring up Rosalind Franklin and Photo 51 and all of that?
You know, I have to say, I really didn't want to even deal with him.
Oh.
I myself didn't want to talk to him. And so my personal interactions with him were limited to things like, would you like a donut? Or you have some coffee? Or, you know, Sir John Gurdon is looking for you. I understand, because I'd heard the stories about people trying to confront him. You know, he just kind of like, Rosalind Franklin, no. You know, he doesn't. It is a really good question. I think a lot of the time when I've tried to challenge people on their narrowness of view, which is something that I have to say, not very good at, and I am working at getting better at. You can watch people deflect. You can watch people kind of like, I'm not going to answer that. She was part of it, but I think the answer would be, what I would expect to hear from him, let me put it that way, is she provided some important data, but she didn't contribute to the conceptual, the sort of the insight of, DNA structure that, Francis Crick and I came up with. That's what I would expect. Like, that's often what people have historically done with the contributions of individuals who were viewed as marginalized, whether they're marginalized by virtue of race or ethnicity or by gender, is, yeah, they're generating data, but the big thoughts are, they weren't a part of that. There's all kinds of weird ways of stratifying that people do to protect their little domain.
Yeah, you, Dr. Sater, you are not the first person that I've heard that's spoken like negatively about Dr. Watson. Like countless videos, just his demeanor and his like outlook on certain things are a little questionable. And it's kind of sad because he's like the face of like, the big discovery. So, or one of the two faces. So I just think it's funny to hear your dialogue on that.
Well, I think this also brings up another point, which is we are taught these narratives about people in science. Science, fundamentally a human thing, right? It's a human activity. There's an interesting story about the people involved. But sometimes the narratives, like any narrative, they will foreground some people and they will erase others. And so one of the things that I've been excited about over the last couple of years is seeing more students ready to challenge those narratives. Who are the people whose work is not being acknowledged? If I can tell a quick story, there's a woman in Germany named Jani Nusslein-Vallard, and she is the person who really launched the new era of Drosophila developmental genetics. And she did this as a postdoc. She talked to her postdoc advisor she wanted to do this giant genetic screen, and he said, "Not in my lab, you don't. If you want to do that, you need to go find your own job." And she took a faculty position at a little tiny university in Germany. And she teamed up with another person, and they did the screen, and it changed the face of the field. It just transformed developmental biology, not just Drosophila developmental biology, but the whole field. And she won the Nobel Prize in 1992. And so this is kind of a mythic story among developmental biologists because, it's plucky Jani Nusslein-Vallard against her, pitted against her advisor, and she prevails, right? So we all love that story, but undoubtedly, there are other sides. And so when we hear these kind of human stories, it's important also to ask questions. And I think the Rosalind Franklin story is a great example of that, but there's a lot more. So, it's always good to have a little skepticism about these mythic narratives.
Hey everyone, thank you so much, Dr. Sater. That's a great way to end presentation with the Nobel Prize. So thank you so much for your time. You gave us a lot to think about. We really appreciate your presentation and your time. Thank you, everyone. Thank you.
My pleasure. Take care.
Take care. Bye.
Thank you. Thank you.
Thanks, everyone.
Bye.
Bye. Stay
Thank
safe.
you.
Thank you. Bye.
Bye.
Thank you, guys.
Bye, everyone. Have a good one.
Have a good morning.
Dr. Gonzalez, I have a question on where we should send our form.